Hello guys, welcome back, and in this video lecture we will talk about antigen-antibody reaction. So when an antigen meets antibody, this reaction can be of two types. Either there can be precipitate formation, so that is known as precipitation reaction, in which a soluble antigen, in here antigen is soluble, and then it combines to the antibody and the precipitates are formed, and this is known as a precipitation reaction. So a precipitation reaction in which the antigen here is soluble, then it forms the precipitation formation. Another kind of reaction that is seen is agglutination reaction. Here agglutinates are formed, clumps are formed, and this agglutination reaction is because of this insoluble antigen. Here the antigen is in the particulate form. So particulate form of antigen combines with the antibody and it forms the agglutination, that is known as agglutination reaction. So when an antigen reacts to an antibody, the reaction can be of two types. Either there can be a precipitation reaction or there can be an agglutination reaction. Precipitation reaction is because of the soluble antigen. Here the antigen is soluble. When a soluble antigen reacts to the antibody, it forms the precipitation. And when an insoluble antigen, that is an particulate antigen, reacts to the antibody, that is known as agglutination reaction. So we will first talk about the precipitation reaction. The precipitation reaction can occur in two kinds of media. Remember, here the antigen is soluble, so it is going to solute, right? So it is going to dissolve. So soluble antigen, here the antigen is soluble. It can be in the solution form, that is the liquid media, or it can be in the gel media. So there are two medias. Either it is present in the liquid media. Here the antigen is soluble. Remember, precipitation reaction, antigen is soluble, while on the other hand, agglutination reaction, the antigen is insoluble or in the particulate form. So in the precipitation reaction, the antigen is present in the soluble form. Either it is a liquid medium or it is in a gel form. So liquid medium, in the liquid medium, it can be like this, a ring formation. So it is the ring test. When a soluble antigen reacts with the antibody, a ring is formed in the precipitation reaction in the liquid media, that is known as a ring test. While on the other hand, there can be flocculation formation in the liquid media, and that is known as flocculation test. In this, the floccules are formed. You can see the floccules, when the floccules like this are formed, that is known as a flocculation test. So in the liquid media, it can be of two types. Either there can be ring test, formation of a ring. If it is formation of the floccule, we call it flocculation test. Then in the gel medium, it is also known as immunodiffusion. So it can be of two types. Either it can be in one dimension or two dimension, or it can be a single diffusion or double diffusion. That we will talk about further in this lecture. So let's talk about the liquid media, this part first. So in the precipitation reaction, that is the antigen is soluble. First, remember two kinds of reaction. When an antigen reacts to an antibody, there can be of two type of reactions. First is the precipitation reaction, another is agglutination reaction. Precipitation reaction in which the antigen is soluble. Agglutination reaction when an antigen is insoluble or in the particulate form. So precipitation reaction, when it occurs in the liquid media, either it can form a ring or it can form floccules. Or precipitation reaction in the gel media. So first talk about precipitation reaction in the liquid media. That is, the first test is the ring test. In the ring test, a ring is formed when an antigen reacts with the antibody in the liquid media. A ring is formed and that is known as a ring test. So in the ring test, the examples are streptococcal, Lancefield grouping. We do it for beta hemolytic streptococcus. This we have covered in the bacteriology. Or for anthrax, there is also a precipitation test, and that is known as S. coli thermo ring precipitation test. In this, the name itself tells that it is a precipitation test. Precipitation test means here the antigen is soluble. Another thing in the name itself tells about the ring. So ring is there. That means example for a ring test. Ring test is a precipitation reaction when it occurs in the liquid media. We have two kinds. Either it can be ring or flocculation. So it is an example for precipitation reaction in the liquid media. It is ring test. S. coli, A for S. coli and A for anthrax. So this test is done for the Bacillus anthrax. S. coli thermo ring precipitation test. We have a hypothesis that is known as Marek lattice hypothesis. In this hypothesis, see, when an antigen reacts with an antibody, so there is a formation of a ring. But before this and after this also a phase is there, and that is known as Merak lattice hypothesis. Merak lattice hypothesis says that when antigen equals to antibody, then there is formation of a ring. So the volume of antigen, when it equals to the volume of antibody, that is the formation of a ring, and this zone is known as zone of equivalence. Zone of equivalence tells you that antigen is equal to antibody. As the name suggests that it is zone of equivalence, equal. Then before this zone of equivalence, there is a prozone, and after this zone is a postzone. So in the prozone here, there is excess of antibody. So here in the prozone, antibodies are in excess as compared to antigen. You can see here the antibody are in more quantity. This also you can see antibody are more, antigen are only 3, while on the other hand, there are a lot of antibodies. Then there is a postzone, post to the zone of equivalence, and in this the antigen are more. You can see the antigen are more and antibody are only few. So this is postzone. You can see more of antigen and less of antibody, and this is known as postzone, wherein the antigens are in excess. So in the pre-zone and prozone, the antibody are more, while on the other hand, in the postzone, antigen are more. So antigen are more in postzone and antibody are more in the prozone. This is the ring test. In the ring test, the ring is formed in which zone? It is the zone of equivalence which forms the rings. This is Merak lattice hypothesis. According to this hypothesis, the ring is formed when antigen equals to the antibody. Coming to another test in the precipitation test in liquid media, it is flocculation test. So there are, first of all, in the flocculation test, when the antigen combines with the antibody, the complex of this antigen-antibody complex, it floats or it is suspended as the particle that are known as floccules. So this is basically, these floccules are basically antigen-antibody complexes. When they float, it seems like these are the floccules. So these are basically antigen-antibody complexes. Either this flocculation can be in the slide or can be in the test tube. If it is done on the slide, it is known as slide flocculation test. And example of this test is known as VDRL. If you remember, the test that is done for Treponema pallidum, the most common test done for Treponema pallidum, which is VDRL for syphilis. Then another is the tube flocculation test. When it is done in a test tube, it is known as tube flocculation test. And again, this is a test for syphilis, which is test for Treponema pallidum, and it is known as Kahn test. So VDRL, the most common test done for syphilis. For Treponema pallidum, it is the slide flocculation test. This flocculation test, when done on a slide, it is known as slide flocculation test. When the flocculation test done in a test tube, it is known as tube flocculation test. We have finished with precipitation reaction in the liquid media. In that, we have covered two things. One is the ring test, another is the flocculation test. Now coming to precipitation reaction in the gel medium. Here the medium is gel, and I told you it is of two types. Either it is one dimension or two dimension. This is one type. Another type, it can be single diffusion or double diffusion. So we are going to cover all of these one by one. So precipitation reaction in the gel medium. Gel which we are taking is 1% agarose gel, 1% agarose gel. Agar we are taking 1%. This is the gel medium that we are taking. Either it can be single diffusion or double diffusion, or it can be a single dimension. One dimension is when it is done in a test tube. Two dimension is when it is done on a slide. So test tube, remember, T for test tube, it resembles the letter 1, and S for slide, and it resembles the letter 2. Basically on the slide, you can see in the 2 dimension. This is how you can remember. Or another way to remember is S resembles 2. So the S is the 2 dimension. If the reaction is done on a slide, it is 2 dimensional. If the reaction is done in a test tube, then it is known as 1 dimension. Now the topic of single diffusion and double diffusion. So single diffusion is when only the antigen moves. This is an antibody. When only the antigen moves, then it is single diffusion. When both antigen and antibody moves, that is known as double diffusion. So single diffusion means diffusion occurs only from the one side. So that is only the antigen side. When the antigen comes and moves, that is known as single diffusion. When both antigen and antibody moves, that is known as double diffusion. Now, We'll talk first about the single dimension. One dimension. One dimension is when the reaction occurs in a test tube. So one dimension. In the one dimension, either there can be movement of antigen only, that is known as single diffusion, or there can be movement of both antigen and antibody, that is known as double diffusion. So in the one dimension, you can remember OOTD. This is the Instagram hashtag outfit of the day. So OOTD, you can remember OO is Ordine test. See, this is Ordine test, and the other test is Oakley-Full-Thorpe test. So Oakley-Full-Thorpe test is done for double diffusion. Single diffusion is Ordine test. Ordine test, if you see in this, only the antigen moves. You can see this is the antigen and there is only movement of antigen. Single diffusion, I told you, remember antigen moves. And when antigen and antibody both moves, then it is known as the double diffusion. So single diffusion example is Ordine test and double diffusion example is Oakley-Full-Thorpe test. So you can remember O for Ordine test, O for Oakley-Full-Thorpe test, and these are the single diffusion and double diffusion. T is our tube and D is dimension. So this is OO, one dimension. Two dimension is, again, two dimension is we have covered when it is done on a slide, when the reaction occurs on a slide. So either two dimension can be single diffusion or double diffusion. Single diffusion is when only the antigen moves. Double diffusion is when both the antigen and antibody moves. So single dimension is also known as the radial immunodiffusion. So here you can see the pattern or the formation is like a radial, is like a circle. And the agar contains the antibody and the well contains the antigen, and here only the antigen moves. You can see this is the agar plate. It contains the antibody and the antigen is in the well. So only the antigen moves, and the antigen antibody reaction shows the precipitate. So here, because it is example for precipitation reaction. So the precipitation here are of different diameters. You can see based on the diameter also it gives the concentration of this antigen. Since there is different diameter precipitations that are formed, you can see there are different diameter. This one is thicker, this one is thinner, this one is more thinner. So based on this, it can tell about the concentration and the substrate class of different antigens and the antibody. So this reaction is known as the Mancini reaction, Mancini method. This is the Mancini method that is for the single diffusion. That is also known as radial immunodiffusion. It is example for diffusion, and here you can see in circles. So precipitate reaction forms a ring. That is why radial immunodiffusion. Mancini diffusion, Mancini method is for single diffusion. Now coming to double diffusion. In double diffusion, both antigen and antibody moves. And here this example is known as outer-Loney double diffusion test. In outer-Loney double diffusion test, there is an agar that contains the antibody and the well contains the antigen. And here you can see different antigens. So they start showing arch. You can see if they start showing arch, that means the precipitate is formed. That means the antigen are identical. The different antigens you are taking, if they are identical, then they are going to form the arch. You can see here the arch is formed. So this is when the antigen are identical, the formation of arc is there. Formation of spur, you can see this is formation of spur in which there is partial identification, partial identical antigens. And here non-identical antigen will show a cross. So since the antigen are not matching, so they show a cross. So different antigen, if they show cross, means they are not identical. The antigens are not identical. And this is seen in the ALK precipitation test that is done for Corynebacterium diphtheriae. So here the agar contains antigen, the agar contain antibody, and the well contain antigen, and both of these moves. Double diffusion, guys, antigen, antibody, both moves. And this is ALK gel precipitation test that is done for Corynebacterium diphtheriae. So remember double diffusion, and the test here is outer-Loney test. So this outer-Loney double diffusion test sounds DD. Lot of DDs are present. And if you remember, DD, that is a cartoon in the Dexter's lab. She breaks everything in the Dexter's lab. So D and D, remember D, D. So double diffusion, 2D. This is example for 2-dimension double diffusion. And here D, D, E, D, D. If you write a D, D, it will be D, D, double E. So it is the ALKS gel precipitation test. That is an example for this double diffusion. So if arch is formed, that means the antigen, different antigens you are taking, they are identical if they are showing arch. If they are showing cross, that means there is no identical antigens. The antigens are not similar. That means they will show cross. They intersect. And if they show spur, that means there is partial identical antigens present. Now, since you have taken antigen and antibody and then you wait for this arch formation or ring formation or whatever it is a time-taking process, right? So to fasten this process, we have come up with an way in which you react the antigen to antibody in an electric medium. So that is electro-immunodiffusion. Here, electric current is passed. Antigen reacted with antibody in a diffusion medium. So here antibody is added to the agar plate and antigen is added to the wells. Here antigens are added to the wells and then you pass the electric current. Once you pass the electric current, fastly it will show the reaction. So it is a faster method. So current is passed only on the slide, guys, and only the antibody moves in one direction. So here antibody moves, not the antigen. Here the antibody moves. So this is electro-immunodiffusion. We have come up with a more faster way in which both the antigen and antibody moves. Here only the antibody moves. In electrodiffusion, you can see antibodies are placed here. Antibodies are placed here. Antibodies on the agar plate. And antibody moves towards the antigen. But counter-immunoelectrophoresis is one in which electric current is passed, but both the antigen and the antibody moves. Now you can see this ring that is formed or the precipitation that is formed is much faster. So precipitant line that is formed is much faster. So it is more faster way in which electric current is passed. In counter-immunoelectrophoresis, electro word means electric current is there. Immuno means antigen-antibody reaction is there. And counter means both the antigen and antibody, and charge is created between both antigen and antibody and they diffuse at the same time. So it is much faster way. Then to come up with a quantitative assessment of antigen, quantity, we want to measure the quantity of the antigen. For this, we have rocket electrophoresis. So in this, antigen is placed in an increasing concentration. So here the concentration of antigen, we keeps on increasing the concentration and antibody is added to the agar. And you can see the rockets that are formed are much more in case of more concentration of antigen. So if you put more concentration of antigen, then the rockets keep on increasing. So this is the precipitant arc that are formed in forms of rockets. So this is for a quantitative assessment of the antigen. Now we have finished with the precipitation reaction. We have covered that there are two kinds of reaction. One is the precipitation reaction. Another one is the agglutination reaction. We have finished with the precipitation reaction. The precipitant rings are formed, floccules are formed. All of these are kind of precipitation reaction in which the antigen is soluble. Now coming to agglutination reaction, wherein the antigen is not soluble. Antigen is in the particulate. So agglutination reaction is better than the precipitation reaction. We have direct and indirect agglutination reaction. Direct agglutination reaction means antigen is going to combine to the antibody and it will react and form the agglutinate. Indirect reaction is in which the antigen and the antibody, first the antigen are sensitized and then it is reacted to the antibody. This is known as indirect. See, direct is the one in which antigen directly reacts to the antibody and they will form an agglutinate. They will form an agglutinate means they will form lumps. While on the other hand, indirect is the one. See, here agglutination reaction, the antigens are insoluble. So first we make the antigen soluble And then we combine the antigen to antibody, and that is known as indirect or passive agglutination reaction. So direct agglutination reaction can be of slide, tube, and microscopic. When we see it on a slide, that is known as slide precipitation reaction. When we see in a test tube, it is known as tube agglutination. And when we see under a microscope, that is known as microscopic. Example one is MAT. So we'll cover all of this. Then indirect agglutination reaction is one in which we are going to first make the antigen soluble and then react it. So direct agglutination reaction is antigen is added to the precipitate antibody, and directly there is an agglutination reaction. Agglutination is done directly. When it is done on a slide, it is known as slide agglutination reaction. For example, we do blood grouping. Tube agglutination reaction, lot of tube agglutination reaction. For example, Vidal test done for typhoid, SAT, that is standard agglutination test, CAT, that is cold agglutination test, Wheelflix test, that is done for rickettsia, Paul Bunner test, that is done for Epstein-Barr virus. All of this we have covered in the micro. Coombs test, and remember microscopic agglutination test is under the microscope. Example is MAT, microscopic agglutination test. So if you can recall from the previous lecture, what we have studied is cat sat on a mat, and this is my billy. So cat is cold agglutination test that is done for mycoplasma. SAT is standard agglutination test that is done for brucella, and MAT is microscopic agglutination test that is done for leptospira. So cat sat on a mat, that is our mnemonic to remember these things. Then we have Coombs test. See, for Coombs test, either it can be direct Coombs test or indirect Coombs test. So in the direct Coombs test, this is done for fetal sample. We take fetal blood sample, we take fetal antibodies, in vivo sensitization for autoimmune hemolytic anemia we do. So basically these fetal cells, these fetal RBCs, we take the fetus blood sample, and these fetal RBCs, they are coated with the antibody in case of erythroblastosis fetalis. And then we react it with the Coombs reagent, and that is known as anti-human globin. Anti-human globin is what? It is the Coombs reagent. When you react these fetal RBCs, you take fetal sample, take fetal blood sample. This fetal blood samples has got fetal RBCs that have got coating with the antibody. And when you react with the Coombs antigen, and this antigen is anti-human globin, and you can see these gaps are filled with anti-human globin, and there is an agglutination reaction. If it comes positive, that means the child is having erythroblastus fetalis. This is direct Coombs test. Indirect Coombs test is done for maternal antibodies for the mother. You take the mother sample to find out if the mother is having antibodies. So it is done for maternal antibodies. Again, in vitro sensitization. This is for fetus, it is in vivo, guys. It is in vivo. While on the other hand, for mothers, it is in vitro sensitization. We take the mother blood sample that has got antibody serum. So we take this, we add anti-human globulin to it, and then the mother's RBC, and if agglutination is seen, if it comes positive, that means the mother is also having. It is also done for autoimmune hemolytic anemia. Coombs test is also done for autoimmune hemolytic anemia. So this is direct and indirect Coombs test. So direct Coombs test is the one in which we take the fetus sample. Indirect is the one in which take the mother sample. This was direct agglutination reaction. Then comes the indirect agglutination reaction. See, in the direct agglutination reaction, these are the examples, guys, for direct, in which we directly take the particulate antigen, react with the antibody, and you can see the agglomerulate. Basically, agglutination is seen. Agglutinates are seen. These are the direct agglutination. Indirect agglutination is you take some particle, and because the antigen here is insoluble, you are going to make it soluble. You take something on which you are going to add the antigen, then combine it with antibody, and then the agglutinates are formed, and that is known as indirect. So indirect agglutination reaction is the one in which the soluble antigens and soluble antibodies are converted to particulate antibodies. So these are the soluble antigens. You are going to put it on a particle that is known as sensitized particulate. On this particle, the antigen is going to come and sit, and then you're going to combine it with antibody, and then you can see the agglutination reaction. This is known as indirect. Here, you are going to take some particulate matter. So that can be, it can be absorbed on RBC or it can be on the latex particulate. So basically latex agglutination reaction in which you are going to take latex particles. So polystyrene latex particles are the one you are going to take. So these sensitized particulate are the latex particles. Latex, polystyrene latex particles are the one can absorb the antigen, not absorb. They are not absorbing it. They are just adsorbing it. They are not taking it inside. They are just adsorbing the antigens, and then you can see the agglutination reaction. So basically some particulate you are taking, and that is known as indirect agglutination reaction. Example is anti-streptolysin O. Coming to another agglutination, that is complement fixation test. In the complement fixation test, we have two tests. One is the Wassermann test or the Treponema pallidum immobilization test. Both are done for Treponema pallidum. Both are done for syphilis. Wassermann test is not done any longer. And the Treponema pallidum immobilization test, TPI test, it is the most specific test for Treponema pallidum immobilization test. Here the principle is antigen combines with antibody, it forms a complex, and this complex is fixed. You can see antigen antibody complement is formed. The complement, it is fixed. So here antigen antibody complex is complemented or it is fixed. This is known as complement fixation test. Now, when you take antigen combined with antibody, this complex is fixed if RBC are not lysed. So as there is no complement for lysis, if RBC are not lysed, that means there is no hemolysis. Here there is no complement, and that means the RBCs are intact. The patient had antibody. And if the RBCs are lysed, that means RBC are broken. Hemolysis is seen. This means that the patient did not have antibody. So there is no antibody. This is how we do the test. If RBC are lysed, they are intact, patient had antibodies. If RBC are not lysed, RBC are intact, that means patient had antibodies. If RBCs are lysed, patient did not have antibodies. So this is how we do the test. This is the complement fixation test. Then next test is neutralization test. Neutralization means we take the toxin, we take the antitoxin, and it is neutralized. For example, for snake bite, you'd give the antivenom, antitoxin. So that is neutralization test. Toxin plus antitoxin, that is going to neutralize it. Examples are Sheik test, that is done for Corynebacterium diphtheriae. Another test is the Nagler reaction, that is done for Clostridium perfringens. Both are done in micro. Coming to very important test, that is ELISA, enzyme linked immunosorbent assay. As the name suggests, there is going to be linking of some enzyme. It is going to be an antigen-antibody reaction. Immuno word is there. Sorbent means it is going to absorb or something. Assay is some test. So it is an enzyme-linked test, right? Which is the enzyme that is linked to this? It is HRP, that is horseradish peroxidase. This is ELISA, that is enzyme-linked immunosorbent assay. So in this ELISA tray, we have some wells. Some wells are present, depressions are present on which we put the sample. So basically you can see 1, 2, 3, 4, 5, 6, 7, 8. 8 this side and 12 this side. So that makes it 12 into 8. That are 96 wells are present, and therefore 96 tests can be done at a time in ELISA. What is the substrate that you are breaking? It is peroxide. And that is why the enzyme peroxidase enzyme is to break the peroxide. So the substrate here is hydrogen peroxide. And in this, color change is seen. And which is the agent that is done to change the color? What is the chromosome? Chromogen, that is tetramethylbenzidine. Tetramethylbenzidine is the one that is going to show the color change. So substrate, basically hydrogen peroxide is broken with the help of enzyme, that is horseradish peroxidase, HRP enzyme, and the color changing properties because of the tetramethylbenzidine. What are the different types of ELISA? We have 4 types of ELISA. Direct ELISA, in which we are going to take the antigen detection Indirect ELISA, antibody detection. In fact, it is the only ELISA that is done for antibody detection. Rest all antigen detection. So direct ELISA, antigen detection, sandwich ELISA, antigen detection, competitive ELISA, antigen detection. The only one for antibody detection is the indirect ELISA. So direct ELISA in the one, see, these are the different kind of ELISA. Direct ELISA in the one is the one that is done for antigen detection, we know. Patient sample, basically antigen that contains the patient, that is in the patient sample, is added to the ELISA tray. And antibody with the enzyme. So this is the antibody here, and here is the enzyme. So this is the antibody with the enzyme. Enzyme is labeled. So this is the antibody. This is the antigen. Antigen is taken from the patient sample. This is the blood sample of the patient. And we have antibody along with the enzyme. This enzyme is known as HRB is added, and the antigen antibody reaction is done. And here we have a substrate. Here we have a substrate. This substrate, when it combines with the enzyme, substrate is what? Hydrogen peroxide. When it is combined with the enzyme labeled antibody, the enzyme is going to break this hydrogen peroxide because the enzyme is peroxidase, horseradish peroxidase, this HRP enzyme. When it breaks the substrate, that is hydrogen peroxide, it is going to show the color because we have the substrate along with the tetramethylbenzidine. So the substrate contains hydrogen peroxide plus chromogen, that is tetramethylbenzidine. So basically, when you are going to react the substrate to the enzyme labeled antibody, antibody with enzyme, when you are going to react it, it is going to show the color. If it comes positive, that means the intensity of the color is directly proportional to the antigen concentration. So if it shows color, means antigen is present. So this is direct ELISA. Direct ELISA is when the antigen combines with the antibody. This antibody has got enzyme along with this HRP enzyme. And then we have a substrate here. This substrate contains what? It contains the hydrogen peroxide. Along with that, it contains the chromogen. What is the chromogen? Tetramethylbenzidine. When this substrate react with this HRP, it is going to change the color. And we see the antigen-antibody reaction. So this reaction is pretty much visible when the colors come out. We know that antigen has reacted to antibody because there is change in color. And this is the direct ELISA test. Then what is indirect ELISA test? Remember, I told you, indirect ELISA is done for antibody detection. Rest all the ELISA are for antigen. Indirect is for antibody. Here, you remember for direct ELISA, this is the antigen. Antibody is coated with the enzyme, and this is a substrate. This substrate is going to change the color. This is direct ELISA. Indirect ELISA is the one antigen is added from the patient sample to which the, see, antigen here is present on the tray and antibody is taken from the patient sample. It is done for antibody detection. So we are taking antibody from the patient here. So antigen is added to which antibody is taken from the patient sample and to which another antibody is added. A second antibody is added. You can see this is the antibody from the patient. A second antibody is added in this sample, and this antibody contains what? This is an enzyme complex, HRP. A second antibody is added. This contains the HRP, the enzyme, and to which substrate is added. This substrate contains what? It contains hydrogen peroxide and also tetramethylbenzidine, which is the chromogen. When it reacts to this secondary antibody, which contains the HRP, it is going to show the color. If color comes, means antibody is present. Here it is for antibody detection because we are taking antibody from the patient sample. So it is done for antibody detection. Intensity of the color is directly proportional to the antibody concentration. This is indirect ELISA. What is direct ELISA? We take the antigen from the patient. Antibody is with the enzyme. Then we have the substrate. This is the substrate. Substrate contains hydrogen peroxide. When we add the substrate, when antigen-antibody reaction occurs because the enzyme is present on the antibody, then it is going to change the color. Indirect ELISA is, we have antigen. From the patient, we have taken antibody. To this secondary antibody is added, and the secondary antibodies contain HRP or enzyme. and then substrate is added. When this reacts, the antigen antibody reacts, then it is going to show the change in the color. So this is indirect ELISA. Basically in indirect ELISA, first of all, we do the antibody detection. That is a different thing. Another thing is we take a secondary antibody. This is the difference. We take the secondary antibody. Then comes the sandwich ELISA. What is a sandwich? We take two pieces of bread and we put something inside, that is known as sandwich. In this sandwich ELISA, we are going to take antibody, both the sides, and antigen in between from the patient, because sandwich is done for antigen detection. Remember, only in indirect ELISA, which is for antibody detection. Rest all the ELISA are for antigen detection. So here antigen is taken from the patient blood and antibody is taken or put on the both the sides. So it is done for antigen detection. So to an antibody, we are taking antigen from the patient sample and we are putting it inside. So this is antibody, and in between antigen is placed, and that is known as sandwich ELISA. Antigen is placed on both the sides of antibody. One antibody, another antibody, and here antigen is placed. On the top of this, we are going to add another antibody which contains our enzyme HRP, and then substrate as added, substrate and tetramethylbenzidine is added. If the color comes, that means it is positive, antigen antibody reaction is there, and more the color means more the antigen concentration, because it is done for antigen testing. Only in indirect ELISA we do the antibody testing. Rest all ELISA are done for antigen detection. Now coming to the competitive ELISA, here there is a competition. You can see it is competing. It is competing. And this is for, again, for antigen detection. I told you, antigen detection. In this, we are going to take the antigen and we are going to react it with antibody outside. Before putting in a tray, ELISA well, we are going to react both the antigen and antibody outside. So first we have antigen antibody complex that is coming from the outside. Then another antigen antibody complex we are going to react in the well of the ELISA tray. So this is competitive ELISA. Basically, what we were doing till now was everything, this reaction was occurring in the wells of this ELISA. This is the ELISA trays. And in this tray, this antigen antibody reaction was occurring. But in competitive ELISA, already there is antigen antibody complex, you can see. That is, this complex is what we are reacting it outside the well. Inside the well, another reaction is occurring, and that is why a competition occurs between both of them, and this is competitive ELISA. You know, this is something like you have studied prior to the class. Prior to the lecture, you have already studied the class and then coming to the class. So this is like a competitive ELISA. So in the competitive ELISA, first of all, antibody reacted with antigen, it is outside the well. Then on this antigen antibody mixture, you are going to coat the well with antigens. Here antigens are present. You can see this is antigen antibody complex which is taken from outside, and inside these wells it is coated with the antigen. Now we have more of antigen. On this you are going to put the antibody that is with the enzyme, and then you are going to add the substrate. You can see here this is something that you are taken from outside, antigen antibody complex. Then antigen is coated on the well of this tray. Antigen are coated. On this, you have added this antigen antibody complex that you have cross-reacted outside. And on top of that, you're going to add the antibody that contains this enzyme HRP. Now this HRP is going to react with the antigen here. And then you add the substrate for the color thing. Right? So this is competitive ELISA. First, antigen antibody complex is present. You have reacted the antigen to antibody outside. Then the wells are coated with antigen. Now you have inside this antigen antibody complex and antigens. Then you add antibody, secondary antibody along with your HRP enzyme, and then you add the substrate also. So there is going to be a change in the color because you have added secondary antibody and antigen. So you are going to see some color change. But after a few minutes, it is going to stop the change in the color. So if this color comes out positive, If this color comes, more color comes, that means the antigen concentration is less. This means that all the antibodies have gone free. There was little or no antigen and there was no reaction outside. So it was free inside and it shows a reaction in the plate. So if the color comes positive, if more color, that means the antigen concentration is less. So color is inversely proportional here. Till now we were taking it directly proportional. Here because of competition, if the more color comes means that now the reaction is occurring between antigen and antibody. That means, say suppose if to this sample you have taken 5 antigens. Initially you have taken 5 antigens and you have added more antibodies to this sample. So we have more substrate. So 5 antibodies are left. See, 5 antigen reacted with 5 antibodies. We have 5 antibodies that are left. So that means it shows reaction, color has come, little color has come. Now, if color has come, more color has come, that means all the antibodies, here all the antibodies are free. There was little or no antigen because all antibodies are free here. So that means there was no reaction that was outside. When you reacted antigen to antibody, there was no reaction outside and it was free inside and it shows the reaction in the plate. That is why it is showing the color. If no color comes in the ELISA plate, if here no color comes as it is, no color change is seen, that means all the reaction has occurred outside only. There is no reaction that occurs inside the plate. All the reaction has occurred here. No reaction has occurred inside the ELISA plate. Antigen antibody reacted earlier, no reaction occurring in the plate. So that means there is more antigen that is present. So that is inversely related. If intensity of the color is more, if the color comes more, that means the antigen concentration is less. If the color is not changing, that means more antigen concentration is present. So this is the competitive ELISA. In direct and sandwich technique, more color means more antigen. Indirect is related to antibody. But here in competitive ELISA, it is not directly related. It is inversely related. So more color means less antigen. This is the difference between competitive and direct and sandwich ELISA. See, we have studied 4 kinds of ELISA. Direct ELISA, indirect ELISA, sandwich ELISA, competitive ELISA. Indirect is the one that is done for antibody detection. While on the other hand, direct, sandwich and competitive are done for antigen detection. In direct ELISA and sandwich ELISA, it is directly proportional. So the color intensity is directly proportional to the antigen concentration. While on the other hand, in competitive ELISA, if more color comes means less antigen is present. So color is inversely proportional to the antigen detection. Now coming to chemiluminescence immunoassay CLIA. In chemiluminescence, as the name suggests, chemi means chemical, luminescent means light. So this is a kind of ELISA in which you are taking HRP enzyme only. But here it is showing the chemical and the light. So shiny chemical light is end point of this test. And instead of tetramethylbenzidine, what do you add here? We add luminol or acridinium esters, and this is going to show the light thing. Then immunofluorescence. Immunofluorescence is the fluorescent gum, which is the dye that is used in machine for fluorescence. It is FITC, which is fluoroisothiocyanate. This is the fluorescent dye that is used in all the machines for fluorescence thing. So this is immunofluorescent. In this, there are 2 things. Either it can be a direct immunofluorescence or indirect immunofluorescence. In direct immunofluorescence, we do the antigen detection. While in direct immunofluorescence, we done the antigen and antibody both detection. In direct immunofluorescence, we detect the antigen, antibody is added to the antigen, and this antigen is taken from the patient sample. So we take antigen from the patient. I'm telling you antigen detection. That means we are taking antigen from the patient, and to this antibody are added. And if the fluorescent signal comes, that means antigen reacted to the antibody and there is a positive reaction. Indirect immunofluorescent means that if you are doing for antibody detection, that means antibody is taken from the patient and it is reacted with antigen. And if it comes positive, if the fluorescence comes, that means it is a positive reaction. For antigen detection, we are taking antigen from the patient and to this antibody is added. And if it comes positive, that means antibody reacted to the antigen. So indirect immunofluorescent example is FTA-ABS, that is our fluorotryponimal antibodies, detection for antibodies, tryponimal antibodies. So this is direct and indirect immunofluorescence. Coming to the last test of this lecture, which is the CARD test. Now CARD test, we have seen this urine pregnancy test kit, UPT. This is a CARD test. You have seen the CARD test in the COVID also. So COVID, we have seen this CARD test doing this. Card test is basically on this card, we are going to put the serum here, and one line is our control line. That means it is going to be present always. It is a normal line that is present. This line tells about the vitality of the test, validity of the test. So basically, if it is present, that means the card is normal. The card is all right. So it is a normal thing. That is a control line. The other T here, one is C, that is a control line. That means this line is normally present. Another line is the T line. This T line tells about the test line. It tells about the validity of the test. If the test come positive, that means this line is present. If this line is not present, that means the test is negative. So this is a card test. On this test, actually inside, there is a cellulose nitrocellulose strip. So this is a nitrocellulose membrane. Once you add serum to it, the serum is going to flow. And if both the lines comes, that means the test is positive. One line is going to come positive always. So the principle that is asked in the exam, it is based on which principle? It is based on immunochromatography. CARD test is done for hepatitis B, hepatitis C, malaria, COVID, urine pregnancy, and for typhoid fever, we have typhi-dot. So this is a CARD test. So guys, this is about antigen-antibody reactions. All the best.